everyone, this is the Theoretical Doctor and welcome back to my channel. If you want to access flashcards on this topic and other videos, do click the link below. I post weekly videos every Tuesdays and Fridays, so do subscribe for more content. In today's topic, we'll be entering oligosaccharides and polysaccharides. It's the continuation of monosaccharide and disaccharide. So if you've missed out on those videos, do check it out. A little bit about oligo or polysaccharides. Oligosaccharides are small chains and they usually contain about 3 to 14 monosaccharides, whereas polysaccharides are polymers made up by condensation of hundreds of monosaccharide monomers through polymerization linked by glycosidic bonds. So basically, oligo and poly are more than 2, but oligo you have 3 to 14 and poly is way more than that. You literally have hundreds. Alright, so the chains of these oligo or polysaccharide, they can be branched or unbranched, and some examples would be starch and cellulose. Starch and cellulose are found in plants. Glycogen is found in animals. So here are some physical properties. They are not sweet and they are insoluble in water. Now this is different from mono and disaccharide, because mono and disaccharide are sweet and they are soluble in water. So if you were to actually put it in hot water, they will form colloids and they cannot be crystallized. They are compact, osmotically inactive, and can be extracted. So some chemical properties, all are non-reducing sugars. Remember, in monosaccharide, all of them are reducing sugars, which means that when they react with Benedict solution, they form brick red precipitate. But for here, they are non-reducing sugars, so they can't react with Benedict solution and they won't form brick red precipitate. Now starch and glycogen, they can be hydrolyzed, meaning they can be cleaved, they can be cut down, they can be broken down into maltose by amylase. And for cellulose, they can be broken down, they can be hydrolyzed in by cellulase. So these are the enzymes to take note. Now let's start off with starch. Starch, there are two forms, amylose and amylopectin, or amylopectin, depending on how you pronounce it. Okay, so amylose are linear, straight chain, unbranched polymer. Take note, linear and straight, okay? There are no branches. So they are linked by alpha-1 for glycosidic bond. Whereas for amylopectin, they are branched. And I'll show the picture in the next slide. They are not just straight, and they are linked by alpha-1 for glycosidic bond as well, but their branches are linked together by alpha-1,6 glycosidic bond. Now, for amylose chain, they will actually coil, take note, they will actually coil into a helix structure. So, like, you see those strands of DNA, those are helix structure, okay? And they are held by hydrogen bonds. When they form the helix structure, they are held by hydrogen bonds formed between the hydroxyl bonds. And for amylopectin, they form a complex and compact 3D structure. Okay, so before I enter on amylose and amylopectin structures, here's a refresher. This is how the alpha glucose looks like, and this is how the beta glucose looks like. So take note of the position of the hydroxyl in the carbon 1. For alpha, it is below. For beta, it is above. Alright, so amylose. Remember I mentioned that amylose, amylose, they are actually linear, straight chain, unbranched. Linear, just straight chain, not branches, meaning on top there's nothing, below there's nothing. It's literally just one straight line, okay? And they are linked by alpha-1 for glycosidic bond. Alpha because this is an alpha glucose, and here carbon 1, carbon 4, they're linked together. So this is the glycosidic bond, hence alpha-1 for glycosidic bond. Now take a look at amylopectin, okay? Remember that amylopectin is a branched. You see here, this is the addition, this is the branch part, okay? So this part here, the linear ones, are linked by alpha-1 for glycosidic bond, whereas the branch part here, they are linked by alpha-1,6 glycosidic bond. Okay, so for cellulose they compose of long unbranched chain of glucose residues linked by beta 1,4-glycosidic bonds. Okay, take note here, it's beta because the sugar being used here now is beta glucose. Okay, so they are actually long like this. They are not branched, but when you put them together, like this parallel, they will be linked by weak hydrogen bonds, just like the water in my very first video. So the water molecules are actually attracted to each other by weak hydrogen bonds. Same goes for cellulose. They are not branched. Okay, like here, it's clearly seen there is a bond. Whereas for cellulose, there's no bond. It's just, there is a bond, but it's hydrogen bond. It's very weak. Okay, so where do these hydrogen bonds form? It's at the hydroxyl group. Okay, OH here. Okay, so what happens is that this chain together, when they come together like this, they form microfibrils, and these microfibrils is important because it gives the plants their high tensile strength and rigidity. Now moving on to glycogen. Okay, glycogen is found in animals, whereas just now I was talking about starch and cellulose. Starch and cellulose are found in plants. Okay, glycogen is similar to amylopectin but with more branches. Take note of this, okay? But with more branches. It's exactly the same. You have alpha glucose, you have the alpha 1,4-glycosidic bond and the alpha 1,6-glycosidic bond because it's similar to amylopectin. But amylopectin, you have less branches, whereas glycogen, they can go crazy with the branches. Okay, so it is the major storage form of carbohydrates in animals. Like I said earlier, glycogen for animals, starch and cellulose for plants, and it's present in the liver and muscle cells. If you've reached this point, thank you very much. Do subscribe and share this video to those who might find this helpful, and I will see you in the next video. Bye!